to your table, and I want you to copy this down. Here we go. Okay, it's a little bit small, so if you need to scoot up, I don't know why, but no one ever sits up front, makes me feel lonely. Here we go. The structure, bone, is a lever. It's a lever that provides the structural support for movement. Provides the structural support for movement. The muscle is the force of movement. By contracting your muscle fibers, you actually exert the effort required to move the lever. Tendons are the attachment point between muscles and bones. How do you remember that, Stickrath? Tense yourself. Tense. Tendons, muscle to bone, right? Ligaments are bone to bone. Tendons, tense, muscles to bone.
Oh.
Ligaments, bone to bone. And nerves are what's going to stimulate the contraction of muscle and help coordinate your movement. Questions about these structures and their roles? No? I'll give you some time to get it ready, yeah? If you are interested in this topic of muscular movement, consider studying exercise physiology in college. There are majors you can take in college that will allow you to study lots of anatomy and physiology, lots of muscles and movement and how it all works together. One of the cool things about muscle cells is they are actually fused cells that are one single multi-nucleated cell, meaning it's got more than one nucleus to coordinate the movement. Alright, I'm going to move forward. That's objective 1. Let's look at objective 2, the elbow joint. You've got to draw, label, and describe the structures involved in the elbow joint. So in doing that, this image is one of an elbow that I want you to be able to orient yourself. Every time you see an image, you've got to be able to figure out, what am I looking at? Right? The bend here, I want you to look first at the bones. So the bend here, you've got a humerus coming down, extending down, and you've got the radius and the ulna. The radius follows the thumb. So as you twist your wrist, you can actually have the ulna and the radius interplay where they move positions. But the radius is going to always follow that thumb. So this would be an individual that's standing like this. Humerus, radius on top, ulna on the bottom. In looking at that, the humerus is the bone of the upper arm. It's going to be attached to the radius via a ligament. This ligament has a name. It's not posted on here. The name is the radial collateral ligament. Collateral, I always think of like collateral damage, right? Which is innocent civilians being hurt in warfare. In this case, collateral is simply part of the complex of this elbow. So we've got the radial collateral ligament, which connects the humerus and the radius. And we've got the ulnar collateral ligament. Here, they just have it labeled ligament. That's the ulnar collateral ligament, connecting the humerus and the ulna. So now we know how the bones are connected. You don't want bone rubbing on bone. That's not good. It's painful. Right? I just visited my 90-year-old grandfather, and when he stands up, it like makes me want to cry because his knees are like, right? He has worn down the cartilage and synovial fluid, which is there to minimize the friction and prevent contact of bone on bone. So the cartilage is attached here at the base of the humerus. There's also a section of cartilage that runs along the ulna and along the radius. The cartilage is there to absorb some of the impact and prevent rubbing of bone on bone. It's encased in what's called a capsule that has a membrane called the joint capsule with fluid inside this. The fluid is to reduce friction, and it's called the synovial fluid. And someone in first period asked me, when you crack your joints, is it bones, like cracking on bones? No, it's gas that's actually trapped in that fluid that gets released. It's gas being released from the fluid. Okay. Is it bad? There's conflicting studies. I haven't seen anything that shows conclusively one way or the other. Some studies say it will lead to arthritis, other studies say it's independent from arthritis. I haven't seen anything that's clear cause and effect. So we know the bones, we know the ligaments, we know the joint capsule and the synovial fluid. Let's talk about the muscles and the tendons. The bicep runs along the humerus, and it contracts to pull the lever up. So when you're moving the lever of your forearm up, you're contracting the bicep. When you contract the tricep, you're going to extend your arm. So flexion and extension. The tricep is composed of three. Tri means three muscle bundles. Three muscle bundles. Bundles. I keep saying bunders. Bunders is not a word. I'm making up words all over the place today. The bicep, bi means two. Tricep, tri means three. So when dudes are in the gym like, oh, I'm going to get some arms doing curls. Not going to get them that way. Yeah, you'll get development of biceps, but if you really want to add size to your arm, it's the tricep that's the larger muscle. It wraps from the inside around the back. And there's three different muscle bundles there. Bunders. You can't stop saying bunders. Okay. Now, connection. How do you connect muscle to bone? What is the type of connective tissue that holds muscle to bone? A tendon. So you've got the tendon of the bicep, which connects the bicep muscle to the radius. When it contracts, it pulls the radius up. When it relaxes, it allows the radius to fall back down. The thing with muscle fibers is they contract and relax. They contract and they relax. They don't push apart. They pull together and then they relax back apart. They pull and they relax. So they're going to be used in opposing pairs. When you flex the bicep, the tendon is going to draw the radius up, and you're going to, at the same time, relax your tricep and allow the movement of the arm. When you're going to extend your arm, so like a lot of swimmers in here, right, when you're doing breaststroke, you extend your arm to push yourself against that water. You're going to extend with the tricep. There is a tendon that joins the tricep to the ulna. It's going to draw the ulna back, and you have to relax your bicep.
So on like this is your recovery. Your bicep is relaxed as what is?
What's the recovery? This motion is recovery?
This.
Oh, drawing it back up. Yeah, so there's a lot that goes into this, right? You can have full contraction of the bicep and relaxation of the tricep, full contraction of the tricep and relaxation of the bicep, or you can have intermediate phases, right? Where you've got contraction of some of those muscle fibers within the bundles. So during that recovery phase, you're going to have biceps engaged and some tricep engaged. Because you're going to actually pull it forward. And if you look at that as a freeze frame over time, you'd be able to tell that. Question and question, yes?
Wait, I'm sorry. So the tendon of the tricep contracts to...
It doesn't contract. The muscle contracts.
The muscle contracts.
The tendon just connects. So the tricep muscle contracts, and in doing so, it's going to shorten. Contraction means shortening. So when the tricep shortens, it's going to draw the bone here, the ulna, back because it's connected to the ulna by the tricep tendon. Okay. Make sense? So right here, you got a tricep tendon that's connecting to this bone. When you flex your triceps, it's actually pulling that bone by shortening back here. And that shortening bunches up, and that's where you get people with the bulging muscles, right? The bulge is the contraction, the shortening of those muscle fibers. Question?
So, if you were to draw nerves,
yeah. Oh, that's a great question. Where would the nerves connect? Whew, it's a tough one. So, absolutely, nerves are going to connect to the muscle fibers, and I'm going to show you how an action potential that we just studied in nerves can be transmitted across a synaptic gap to a muscle cell and stimulate the contraction of that muscle cell. That's an animation I'm going to show you in a little bit. So there's absolutely nerves around the muscle tissue. In terms of the bones and the ligaments and so forth, I'm not sure. I need to look into it. It's a good question. The nerves are going to coordinate the contraction of the muscles, and whether or not those are connected along the elbow joint to the various types of tissue, I need to look. It's one that I should look into. Send me an email so I can have the answer for you by Monday. All right. That's it for this diagram. Other questions about the diagram? Looking back to the objective, one of the things, just so you know, Alec, the nerves are involved up here, like what's the role of nerves, but they're not expecting you to be able to put them in a diagram. I'll be happy to share that info with you, but it's not part of this objective. Okay? However, look at objective 3. You're going to outline the function of those structures. That's a combination of 1 and 2. What's the function of the radius? It's a bone, it's a lever. What's the function of the bicep? It's a muscle. It's going to provide the force that's going to move the lever. Right? What's the function of the nerves that are going to connect to the muscle? Stimulate contraction and cause movement. Right? So you've got to combine 1 and 2 in your understanding of 3. Questions for me? Because the image that I have associated with 3 is just the same image. You've got to be able to explain the function of each thing. What's the function of the synovial fluid? Reduce friction. What's the function of cartilage? It's a type of tissue that is going to protect the bone from damage by rubbing bone on bone. Yeah?
So you got this little sack with
Yes.
synovial fluid, right?
Yes.
And the cartilage is exposed on the bone.
Right.
On the end of the bone,
Yes.
inside the sack.
Yes.
Inside the fluid.
That's correct. That's correct. Everything you just said is exactly right. We'll move on to the next objective. What's the membrane for that joint capsule made out of?
Yeah.
I need to look. It's going to be some type of connective tissue, but I need to look what it is.
Could you,
Damage it?
yeah.
Absolutely.
Displace them both.
Yeah. Yeah, you can damage it for sure. Yeah. All right. So let's look at number 4. Compare the movements of the hip and the knee. I'm going to extend this one a little bit beyond the hip and knee. You should also be able to give me another example like the hip and another example like the knee. So let's first go to the type of joint, or the pivot point of the joint. The hip is a pivot joint that functions in many planes, whereas the knee is a joint that functions only in one plane. Let's talk about what that means. Your hip, you can move your leg up, you can move your leg back, you can move your leg side to side, you can bring it around this way, you can bring it around this way. It's a ball and socket joint. So that the top of the femur, the cap of the femur, is a ball shape. And the pelvis has a socket that fits with that ball, and you can actually roll it around in a bunch of different planes. Whereas the knee joint itself is set to move in only one plane. Now you can orient your leg in different directions, like a soccer player, right, in order to move, but the joint itself only moves in one plane, forward and back. Now there are ligaments that get damaged frequently in athletics that are supposed to hold the knee in one plane of movement. What happens when your knee goes sideways? You damage your ligaments, and there's two main ones that get damaged, the ACL and the MCL. A stands for anterior, M stands for medial. It's the positioning in relationship to the knee. The L stands for ligament. I forget what the C is. Somebody Google it and tell me. I don't remember. But they're specific ligaments that support the knee joint so it can move in one direction. When they get damaged, you have issues with the movement of the knee. And it takes a long time for recovery. And generally, those individuals that get damaged have rehabilitation therapy that they need to go through to strengthen the muscles around the knee to provide the support. Now, let's talk about the bones and how this works, and then we're going to give some other examples as well. So this is the minimum. You should be able to say that the hip involves two bones, the pelvis and the femur. The pelvis is the socket. Femur is the ball. The lever that's going to move is the femur. So that's talking about the upper part of your leg moving up and back, up and down, right? When you move the leg up, that's flexion of the quadriceps and relaxation of the hamstrings. Remember, opposing muscle groups. When you're going to extend the leg back, you have flex of the hamstrings, contraction of the hamstrings, relaxation of the quadriceps. When is this most frequently used? Jumping. When you go down to prepare for a jump, you're in a squat position. Your quadriceps are engaged and your hamstrings are relaxed. When you then extend your legs and jump off the ground, you are pulling the hamstrings tight and relaxing the quadriceps. Does that part make sense?
Yeah.
Yeah? The knee joint is a separate type of lever, so I'm going to use this table as an example. When you're working with a knee joint, the tibia is the main bone. It's the lever that extends, it's the shin bone, really. The fibula is a small bone that runs on the outside. It's protected by a lot of muscle, and it's hard to damage the fibula. A friend of mine broke his fibula last year because he ran into a tree when he was snowboarding this direction. Boom. And he snapped his fibula, right? But the tibia is a bigger bone. And it's the shin bone that you protect with shin guards in soccer. It's the one that you can feel right along the leg there. You have muscles that are attached to the shin bone, the tibia, by tendons. And when you flex your quadricep, you're going to extend the leg and pull it upwards. When you relax the quadricep and flex the hamstring, you pull that shin bone back as far as you can towards your butt. All right? You guys got that? Yeah? What?
I mean, why are the bones more exposed, say, at the end of the arm and the shin?
Good question. I don't have an answer. Why are the bones exposed more at different regions of the body? I don't know.
It's like your calf goes up halfway up the leg.
Yeah.
The rest is less.
Good question. Good question. I would say it has something to do with the actual movement of it. If your calf went all the way down to your ankle, there would be a different type of movement there. So you're talking about muscles in terms of protection, but they're not really there for protection. They're there for movement.
Protection is why they
Yeah. Yeah. It has to do with there's a whole physics component of this that you can look into about the length of someone's body and how their muscles function in relationship to their body based on attachment points. Let's talk about some other examples of hinge joints and ball and socket. Give me one other example of a ball and socket joint. Yeah. Shoulder. Right. Your humerus, the top of the humerus. The cap is a ball, and the socket is formed by a combination of the clavicle and the scapula and the shoulder area there. They're attached to each other via ligaments, and there are muscles responsible for moving that, right? You can move your arm in many planes. That's a ball and socket joint. What about your elbow? What kind of joint is your elbow joint?
One.
Which would be a hinge joint. A hinge joint. Not a ball and socket. Have you noticed that it goes ball and socket hinge, ball and socket hinge to your extremities? Yes?
What about like fingers, hinge joints?
Yeah, fingers are hinge joints, right? Like if you crack, if I took your finger and I went, right? It's going to damage that joint. Yes?
What about the wrist?
Good question. The wrist, there's lots of little bones in the wrist, right? And the wrist is obviously more complex than what we need to know for this course. Don't have a good answer for you. Would have to consult an anatomy book. But I can tell you that it's the interaction of a lot of muscles and a lot of bones. Yeah?
Your head connected to your spine, that's not a joint,
The connection of the
right? Like,
cranium
just because
to the
you
spinal
move your
column is, I don't know. Daggone, you're asking good questions. You guys forget, I'm a bio teacher. I'm like jack of all trades, master of none, and these are good physiology questions. I don't know. Someone that has a lot of physiologic background probably knows that. I know this. So we can Google it, check it out. You can email me and I'll look into it. I will agree to find out answers if I don't have the answer, so go ahead. Yeah? Same type of thing? Dude, you guys that love this, you should take anatomy and physiology in college. Okay? You absolutely should. It's an interesting course. All right. Next. Let's talk about striated muscle fibers. So you've got smooth muscle that lines your internal organs, and you've got striated muscle that looks like it's got bands. That's skeletal muscle involved in movement, and that's actually what we eat. When you're eating animals like steak and burgers, you're eating striated voluntary muscle groups. You're not eating those involuntary smooth muscle groups. You could, I suppose. There are some exotic foods that would eat that. But most of the food you're eating is skeletal muscle. All right, so we need to describe this skeletal muscle, including myofibrils with the light and dark bands, the mitochondria, sarcoplasmic reticulum, nuclei, and sarcolemma. All right, let's go through that. A lot of this, they're just different words because they're specific to muscle fibers, but they're ideas you already know. So we're going to look at this as a series of diagrams. Okay, this comes from the site Click for Biology, by the way. Really helpful if you want to check it out. It goes through a lot of the idea objectives in order. So, muscle. That's item number 2 here. Right? This shows contracted muscle. Tendons are item 1 at the top that are going to attach that muscle to the bone. The muscle, if you cut it right down to where the 2 is, straight in half, you would examine and see that they're actually bundles of fibers that are bound together in cylinders. This is what you can see when you're about to eat a piece of meat. You can see the striations. Now, the muscle, the membrane of that muscle cell is called a sarcolemma. Sarcolemma. It's the plasma membrane surrounding the cell. But because we're talking about muscle cells specifically, it has its own unique name. It's called a sarcolemma. The reticulum, we learned it as the endoplasmic reticulum, the rough ER and the smooth ER. The reticulum in muscle cells is called the sarcoplasmic reticulum. So if we were to take out one of these bundles of fibers and look at it more closely, you would see it's multinucleated. All the little black cylinders represent, or black circles represent nuclei. You would see that there are smaller gray fibers in here. Those are called myofibrils. Myofibrils are a combination of two types of protein, actin and myosin. So if we look at one of these myofibrils and we pull it out, you would see actin and myosin. And these are going to be represented in bands. So there's light bands and dark bands we're going to talk about. The actin here is represented in purple. It's a protein. The green is a protein called myosin. And the interaction of actin and myosin is what's responsible for the contraction and relaxation of your muscles. We're going to go into a lot more detail about this with animations, but the basic idea here is these little extensions coming off the green are going to, through the use of ATP, attach to an actin fiber and pull it. That's called dynein walking, and we're going to see a lot on this. But what happens is, picture these purple segments here. When the myosin fibers each grab and contract, it pulls the ends of this band together. Let me show you some other images that'll help make this more clear, and then we'll go to an animation. So looking at this image, the bands themselves are called Z-bands. So the actin filaments are thin filaments that are going to interact at the Z-band with a myosin, which is a thick filament. The Z-band will actually change and become more narrow and wide based on the contraction of the muscle cell. So when the muscle contracts, the Z-bands become more narrow. And when the muscle relaxes, the Z-band becomes more wide. This unit is called a sarcomere. S-A-R-C-O-M-E-R-E. It is an often asked question in IB tests, looking back for the past 14 years. This unit is called a sarcomere. And this is what it looks like under an electron micrograph. Dark band, light band. Okay? And the Z's are going to be changing in size. So, look at all the mitochondria here to supply the ATP necessary for the myofibrils to contract. The myofibrils are just small sections of muscle cells. In order to make it a little bit more clear, let's look at this diagram. Relaxed muscle, the Z-bands are spread apart. Contracted muscle, the Z-bands have become more narrow. So that when you want to contract, you pull together. That pulling together bunches the muscle fibers and draws the lever in a particular direction. Question?
So myosin pulls on actin, which pulls on Z.
Correct.
Okay.
And I'm going to show you some animations. Let's take a look at those now. I've got 5 animations to show you. I won't get them all done today, but I think these are really helpful. And to access them, just go to the website, and this is under the exam review page. I'll put them on the anatomy and physiology page as well. But here, exam review page, these animations, and there's some quizzes that follow. This is from a human anatomy book. So I'm going to start by just showing you the connection between a nerve cell and a muscle cell. Right, so that's where we're going to go first. The yellow here represents a nerve cell. We're looking, as we learned last week, at the end of a nerve cell called the axon terminal, right? So the axon will be up here, and the action potential travels down the axon terminal. When it reaches the terminal, there are calcium channels that will allow calcium to flow in. The influx of calcium, we learned this last week, the influx of calcium causes these vesicles with neurotransmitters to undergo exocytosis and diffusion of the neurotransmitters to receptors. But check this. Instead of one nerve cell to another nerve cell, as we learned last week, it's a nerve cell to a muscle cell. And that nerve signal will transmit the stimulus to contract the muscle fibers. So we've got nerves connected to muscles here in the first diagram. It's the first thing we know. Let's take a look at it.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So that's synaptic transmission of a nerve to a muscle cell. That was pretty fast, yeah. Yeah, but we're going to slow down and talk more about this on Monday. Yes?
So choline goes back into the presynaptic terminal and then binds with acetic acid to make more acetylcholine.
That's right.
Where does the acetic acid that was originally
Good question. I don't know. Yeah. Yeah. So he's like, what happened to the little orange balls? The yellow balls get reabsorbed. What happened to the orange ball? Yeah, I don't know. That's a good question. Others before I move on, I want to show you one more video segment here. The next video segment I'm going to show you talks about the action potential as it's propagated down a muscle cell, how it results in contraction of the muscle. So let's take a listen to this one. Okay, to orient yourself here, we've got a sarcomere that they're showing enlarged. It's an enlarged version to show you what's happening in the muscle cell. Here we go.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell.
All right, let's stop for a second. They said some words there. I want to make sure we know. Sarcolemma, what is that?
the membrane.
That's the membrane of the muscle cell. So we've got an action potential propagating down the membrane of the muscle cell, from a nerve to a muscle cell, traveling down the membrane of the muscle cell to what's called a T-tubule, where it's going to actually interact with the muscle fibers.
So
Near the ends
T-tubules are just entrances.
T-tubules you can think of as entrances.
of the sarcoplasmic reticulum
Back up for a second.
into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament.
Let's stop for a second. So we've got actin on top, that's the purple. The green represents myosin. Tropomyosin is simply the region of the actin filament that's going to react with the myosin filament. Tropomyosin reacts. But it's not part of myosin, it is part of the actin filament. We good on that? Yeah? So this is an expanded view The myosin is always going to be represented in green, the actin is represented in purple.
Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin.
So the calcium influx causes a conformational change in the protein.
When the tropomyosin moves, this uncovers the myosin binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
The last video I want to show you, I know I'm going fast, we'll slow it down on Monday. I want to show you one more about how the myofilament works specifically. So now we've got this myosin is going to bind to the actin, and as it does so, it's going to pull it in one direction.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Question? Yeah. How does it relate to muscles contracting? Good question.
Does that happen?
Let's go back to this. Well, wait, let's go back to this one. So what we're seeing, that's an expanded view, right? So what you're seeing here would be all of these green heads binding to the actin and pulling them together. So they're all interacting in one direction. These are pulling the actin this direction, these are pulling the actin this direction, so that you get this fiber actually shortening. And the shortening of the fiber is what causes the bunching together.
So does it happen really quickly?
Absolutely. Yeah, they're showing this very, very slowly, step by step, right? But you got to figure, you got millions of these that are all interacting, and they're all controlled by nerves and multinucleated cells, so that they have coordinated movement. Yeah.
How does the myosin head know when to stop binding to the actin?
Good question. There's some sort of signal to release it, and we know that ATP is what powers that release. But where it receives the signal and how that all goes, extremely complicated detail. I don't know. We don't need to know for this course. Okay. One more I think will help you, Anna, because it goes and shows this in less than just like the one small image, step by step. I think this will help a bit more. Let me go break down. All right. So here's your sarcolemma. Or I'm sorry, your sarcoma. Not sarcoma, that's a cancer. Sarcomere. This is a sarcomere, right? So we've got the Z bands out here, right? And we've got the actin and the myosin represented in green. So it's going to show you one unit of muscle contracting.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
So as long as calcium ions are influxed into that cell, it's going to continue to contract.
And the calcium comes from the nerve impulses.
It comes from the nerve impulse, that's right. Last one, and then we're done for the day. So there's two of these now, it's two sarcomeres.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
I'll show you this one more time when we're done.
In a relaxed muscle,
Relax.
actin
Right.
and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
You don't need to worry about H zones, I bands, and all that. It is something that, if you're interested in this, you'll learn more about. Yes, go ahead.
So the muscle knows when to stop contraction when the actin is released.
When it reaches the end of the actin, it can't contract anymore.
Oh, when it gets to
Right.
the band.
Yeah, that's right. So that's why there's a limit.